Okay, so today we're diving into something pretty amazing, the unsung heroes inside every single living thing. Enzymes. You can think of them as these microscopic powerhouse machines that are, quietly running the entire show, making life itself possible. But to do that, they have to solve a huge chemical problem.
I mean, really, think about this. In a chem lab, right? You often have to crank up the heat a lot just to get a reaction going. But your body? It's pulling off trillions of incredibly complex chemical reactions all day, every day, at a super gentle 98.6 degrees. So, how on earth does it manage that without, boiling over?
Well, the answer is a team of microscopic superheroes, and we call them enzymes. They're the whole reason life can exist in this delicate balance, pushing forward all these reactions that would normally need just a destructive amount of energy.
So, what is an enzyme really? Well, the official definition is a biological catalyst. And what that means is they're almost always proteins, and their one big job is to dramatically speed up chemical reactions. We're talking millions of times faster. And the craziest part? They don't get used up in the process. They're basically the ultimate reusable tools of biology.
All right, Section 1, Life's Chemical Challenge. So how do these little molecular machines actually pull this off? Let's get right into the nuts and bolts of how they work. Okay, so it all kicks off when a specific molecule, which we call a substrate, finds its way to a very special spot on the enzyme. This spot is called the active site. And this isn't just a random bump in, it's super, super specific. This initial connection, this hug between the enzyme and its substrate, creates what's called the enzyme-substrate complex. And that right there is the first critical step.
Now, how scientists thought this worked has actually changed over time. The first idea was this lock and key model. The enzyme's active site is a rigid shape, and only the perfectly shaped substrate key can fit. But it turns out it's a bit more dynamic than that. The model we use now is called induced fit. This one says the enzyme is actually flexible. As the substrate comes in, the active site literally changes its shape to create a perfect, snug embrace. It's much more like a hand fitting perfectly into a glove.
Okay, Section 2, Meet your body's catalysts. Now, it's really important to remember that enzymes aren't a one-size-fits-all kind of thing. Nope, there's an incredible variety of them, each one a specialist for a different job. So let's meet the team and the helpers they need to get things done. To try and make sense of this huge variety, scientists have grouped them into 6 major classes. You've got transferases, which, just like their name sounds, transfer little bits from one molecule to another. You've got hydrolases, think of your digestive enzymes here. They use water to break bonds. And then you've got ones like ligases that do the opposite, stitching molecules together. It's just, wow. It gives you a sense of the incredible range of tasks these things are handling inside you right now. But here's the thing. As powerful as enzymes are, a lot of them can't do their job alone. They need a little non-protein helper, a sidekick, if you will, called a cofactor. Without it, the enzyme is just incomplete. It's called an apoenzyme, and it's basically sitting on the bench, waiting to be tagged into the game. And these little helpers, these cofactors, they come in 2 main types. First, you have coenzymes, which are organic molecules, and get this, many of them come directly from the vitamins in your food, like vitamin B1. Then you have activators, which are inorganic ions, think minerals like zinc or magnesium. And that right there is the direct link. That's exactly why your doctor is always telling you to get enough vitamins and minerals. Your enzymes literally need them to function.
All right, on to Section 3, a delicate balancing act. Because here's the deal. Enzymes might be powerful, but they are also incredibly picky. You could almost call them divas. They can only perform their miracles under very specific conditions. So let's look at the delicate environmental balance they absolutely have to have.
Every single enzyme has what's called a Goldilocks zone, a perfect temperature and a perfect pH where it works best. Not too hot, not too cold, just right. And you can see it on the chart. If things are too low, activity just kind of slows to a crawl. But if they get too high, the enzyme starts to unravel, to lose its shape. We call that denaturation. And its activity just completely nosedives. And this is exactly why a high fever is so dangerous.
It's literally cooking your enzymes, pushing them out of that perfect zone, and messing up all those critical chemical reactions. The amount of stuff the enzyme has to work on, the substrate, that matters a lot too. At first, the more substrate you throw at it, the faster the reaction goes. But that can't go on forever. Think of it like cashiers at a store. Eventually, all the cashiers, the enzyme's active sites, are busy. They're saturated. At that point, the line can't move any faster. The reaction hits its top speed, what we call Vmax. And a really important number here is Km. That just tells you how much substrate you need to get the reaction to half of its top speed. If an enzyme has a low Km, it means it's really good at grabbing its substrate. It doesn't need much to get going.
Okay, section 4, turning enzymes on and off. Think about it. Having all these super fast reactions going full blast all the time would be complete and utter chaos inside our cells. So how does the body keep it all under control? Well, that brings us to the really crucial topic of regulation.
To keep everything in order, the body obviously needs a way to hit the brakes. And that's exactly the job of something called an inhibitor. An inhibitor is basically any molecule that can slow down or even completely stop an enzyme from doing its thing. It's a fundamental layer of control.
So when it comes to hitting the brakes, there are two main ways to do it. First, you have competitive inhibitors. These guys look a whole lot like the real substrate, and they literally compete for the same active site. It's like someone stealing your reserved parking spot. The only way to win is to just flood the area with more of your own cars, or in this case, more substrate. But then you have non-competitive inhibitors. These are sneakier. They don't go for the active site. They bind somewhere else on the enzyme entirely, and that binding changes the enzyme's whole shape, which messes up the active site. And in this case, adding more substrate does absolutely nothing to help.
And this whole idea, this is a massive deal in modern medicine. So many of the drugs we rely on are, at their core, just enzyme inhibitors. It's brilliant, really. Take sulfa drugs, those antibiotics. They work by competitively blocking a key enzyme that bacteria use to make folic acid, which they need to survive. And penicillin, it's even more direct. It permanently shuts down the enzyme that bacteria use to build their cell walls. No cell wall, and pop. The bacteria basically bursts. It's an amazing application of this principle.
And here's a totally wild fun fact for you straight from nature. Ever wonder how a parasitic worm, something like an ascaris worm, can live in your gut without just being digested? Well, it pulls off an incredible trick. It releases its own powerful, non-competitive inhibitors that shut down your digestive enzymes, pepsin and trypsin. It literally creates its own safe zone. It is a fascinating and, yeah, kinda terrifying example of biochemical warfare happening inside a living body. So there you have it. From digesting your lunch, to fighting off bacteria, to even how parasites survive, enzymes are just at the heart of it all. They are life's master chemists, nature's incredibly elegant solution to some really complex problems. Which leaves us with a big question, right? As we get better and better at understanding and even controlling these tiny machines, what's the next big problem we could solve? In medicine? In creating new forms of energy? It really makes you wonder what's next.